Welcome to Gadlafoy Academy, and in this session we'll be looking into viruses as part of the BGCSE syllabus for biology. So we study viruses as a way of understanding whether they're living or non-living organisms. So there are aspects of them, of viruses, that actually maybe in their structure that show that they could be living organisms, but as we study them more, we understand maybe there are also aspects that also show that they are not living organisms. So when we look into the structure here, we've got this outside layer, which is called the envelope. And the envelope comes from the host cell membrane. And we'll understand this more on how this comes about in the section of reproduction. And then we actually have this, what is called this layer here. This layer here that is called the protein coat. The protein coat, which is normally called the capsid, is made up of subunits called capsomeres. And inside we'll have a coiled strand of DNA, or it could be RNA. So we should understand that viruses have no nucleus, cytoplasm, cell organelles, or cell membrane. And that raises the question, can we then classify them as living organisms? But however, we realize that, they have DNA and RNA strands that are surrounded by the capsid, which we know that these are subunits of living organisms or living cells. Viruses are also very small. With you'd find them when you look through a microscope and measure them, that have 20 to 30 nanometers, and that's quite small. That's like to an order of 9, and they can come in different shapes when you look into them. They are secular. There are those that are secular. And there are also those that could be in coils and stuff like that. Yeah. And then we've got reproduction. And in reproduction, that's where we actually see. So we would have our virus, and we would actually have maybe a normal cell. So what happens is that the virus attaches to the normal cell. And what would happen is that in this process, it is actually absorbed into the cell. So this is process 1, where it's actually absorbed into the cell. And when it is absorbed into the cell, it then uncoils in the cell. The other process is whereby when it attaches into the normal cell, it injects the RNA or DNA into the cell, which should just straightforward lead to the process over here. And from here on, RNA or DNA replication happens. So what actually happens is that the virus actually uses the ATP, the energy inside the cell generated by the normal cell to actually replicate itself, in which it's the cell carrying out the process of actually replicating the virus, and the virus is actually taking advantage of that process in the cell. So when DNA and RNA or RNA replication happens, we see many viruses being produced inside the cell. And when the cell is going to increase in size until it actually bursts out with the viruses being released. And this is actually how you have the HIV virus actually causing harm in T cells, whereby the virus actually invades T cells and causes them to burst in this process. And when you look into the nutrition of viruses, you realize they actually don't feed. There's nothing that actually they don't feed from the host cell. They just live in it, actually use it for reproduction. And when you look into the useful and harmful viruses are used in making vaccines and also in genetic engineering. And they're also harmful viruses actually we know cause diseases like COVID-19. So thank you for joining us in this session.